It is now my pleasure to introduce Jim Hanken, who is the Alexander Agassi Professor of Zoology and Professor of Biology in the Department of Organismic and Evolutionary Biology here. He is Director of the Museum of Comparative Zoology and also Curator in Herpetology. And I should say up front that I've known Jim for many years and seen how much he's been a tremendous and tireless advocate for the value of museums, our collections, and our public programs. And he continues to dedicate an enormous part of his professional life to increasing awareness of the importance of our collections to the many different areas of human endeavor, as we are going to hear about tonight. And for me, one of the wonderful things about coming to Harvard was the opportunity to work more closely with him and to learn a great deal from him. So I consider that a huge privilege. Of course, it's not all about museums. He also does quite a lot of research focused on the evolution, morphology, developmental biology, and systematics of amphibians. And his lab studies the evolution of cranial facial patterns in vertebrates, the taxonomy and evolution of African frogs, Asian salamanders, amphibian conservation, and he maintains field programs in Mexico, China, and Africa. So hence all the traveling, I guess. He was also closely involved with the Encyclopedia of Life, this unprecedented effort to make available information for all species of life on Earth on one website that I'm sure many of you are aware of. He was the chair of the steering committee, continues to serve on the executive committee, and is responsible for the EOL Learning and Education Group, which is headquartered here at the Museum of Comparative Zoology. He received his BA and PhD in zoology from the University of California at Berkeley, and he joined the faculty here at Harvard as professor of biology in 1999 after teaching in the Department of Environmental, Population, and Organismic Biology at the University of Colorado in Boulder. In 2002, he was appointed director of the Museum of Comparative Zoology. He has published extensively and has received numerous honors and awards, including grants from the National Science Foundation, the John D. and Catherine T. MacArthur Foundation, and the Alfred P. Sloan Foundation. His past professional service is far-reaching, including membership on the board of directors for the American Institute of Biological Sciences, associate editor of the journal Zoological Science, president of the American Society of Ichthyologists and Herpetologists, and a member of the board of directors for the American Association of Museums. He actually said to me he hates it when people stand up on the podium and talk for a long time about the speaker instead of getting on to the main event. So I am going to stop right there and allow us to get on to the main event. Professor Jim Hanken, thank you.
Thank you, Jane. Hello, everyone. And let me also thank you for coming out on what is a very cold night. I actually wasn't going to tell you about the Hawaii trip, but now that the cat's out of the bag, I do have a request. When we get to the question session, if you could be brief, please. I need to save time to get home to find my bathing suit and suntan oil. So, in many respects, I'm being a director of the MCZ, and in fact just being from Harvard, am an unusual choice to talk about the role of natural history collections in evolution. And to illustrate this, I'm going to give you 4 quotes, or quotes from 4 different people who have populated this building or nearby for the last over the last 150 years, and I think you'll see what I'm talking about. Let me start with this man. This is Louis Agassiz, who's the founder, larger-than-life figure from the 19th century. He was the founder of the Museum of Comparative Zoology in 1859. He was, in modern parlance, we would say he was a creationist. He was a bitter opponent of Charles Darwin. They had a long-running feud. He never believed Darwin's theory of evolution by natural selection or evolution at all, which was known as the transmutation theory. And in fact, the great irony of the Museum of Comparative Zoology, which has some of the most vast collections of natural history specimens in the world, many of which were amassed by Agassiz, he gathered them with the principal aim that they would ultimately provide the evidence to refute Darwin. And here is a quote. This was one of his most famous expeditions, the Thayer expedition to South America in 1865. He died in 1873. I'm often asked, what is my chief aim in this expedition to South America? No doubt, in a general way, it is to collect materials for future study, but the conviction which draws me irresistibly is that the combination of animals on this continent, where the fauna are so characteristic and so distinct from all others, will give me the means of showing that the transmutation theory is wholly without foundation in facts. Now, one of the people, another character, one of the people on this expedition, William James. This is the same William James of William James Hall. He was an undergraduate at Harvard at the time. He went along with Agassiz as one of his assistants as an undergraduate on the Thayer expedition. Later founded the field of psychology and was hired back as a professor. The idea of building collections, he really wasn't into it too much. If there is anything I hate, it is collecting. I don't think it is suited to my genius at all. Another person, Thomas Barber, much revered former director of the MCZ from 1927 to 1946. Barber did his field work principally in Central America and the West Indies, including Panama, which is where this photograph was taken. Now, Barber, you have to understand, he was a really tall man, but not this tall. The man he's standing next to is not that tall. But Barber came in as the director of the MCZ when the building was already pretty full. And he had the problem of figuring out how are we going to make room for new collections. And one of the things Barber is known for is, as director, if there were specimens that he didn't think were particularly helpful or necessary, he would just tell people to throw them out. But not just throw them out, they threw them down the elevator shaft in the MCZ, the same elevator shaft we still have. And apparently when the pile of carcasses and other things at the bottom of the elevator shaft got so deep, somebody was asked to bring a wheelbarrow or other conveyance and drag them all out and put them out on Oxford Street by the sidewalk for people to take away as they wished. Another example of specimens that we have, which Barber had an interesting approach to, are these drawings. This was a drawing from the Thayer expedition, actually, by Louis Agassiz's artist, watercolorist, Jacques Burkhart, a spectacular artist who made just marvelous representations. He would be on the ship, on the deck of the ship in the Amazon as they were dragging on board all these new species of fishes, and Burkhart would frantically scramble, taking color notes and making sketches and so forth, and when he came back here he would ink up all of these drawings and so forth. And Barber saw them and he figured, well, we don't need these. So he just sent, fortunately he didn't throw them out, but he put them in a box and he sent them to George Myers, who was an ichthyologist at Stanford University. Myers didn't know what was going on. He wrote to Barber and asked, what's the deal with these pictures? And here's what Barber wrote back. I was called away from Cambridge just after the Burkhart sketches were sent to you, and that's why no letter accompanied them. They are a gift, pure and simple, and I hope that you may someday perhaps find some use for them. I suppose even in California you occasionally have a fire in the grate. You'll be pleased to know that they were ultimately returned here post-Barber, and recently were digitized by the Meyer Library, and many of them are available online if anybody wants to download them for free. The final person, not an MCZ denizen, but this is legendary Dean of the Faculty of Arts and Sciences, Henry Rozowski. He was the Dean from 1973 to 1984. And he made an observation about natural history science at a time, and I think it really was in a bit of a low ebb and being eclipsed at the time by molecular biology. Humanists may, and this is from his book that he wrote in 1991, humanists may believe that they have a near monopoly of suffering and neglect. Yet there are branches of the natural and social sciences that also bear relative deprivation. For example, the use of science, these italics are mine, use of scientific museum collections, botanical, zoological, and geological, is no longer required by most leading-edge researchers. Molecular and cellular techniques are crowding traditional systematists, those charged with maintaining these institutions for a future time when they may well be in great demand, fully share feelings of envy. Okay? Not a very proud moment in the history of natural history collections, and quite worrisome, in fact. Now, what I'm going to do today, despite my origins here at Harvard, is the following. First, I'll give you a brief overview of the history of natural history collections, and it really will be brief. It's a very large subject that I can't go into in any detail. Then I'll try to explain to you the fundamental role of natural history collections in the study of evolution, principally in the discovering and naming of species and documenting the diversity and distribution of life on Earth. This is the fundamental set of activities that must be taken before we carry on more higher order and advanced studies of the evolutionary biology of organisms. And here I just want to endorse what Jane said. There are three excellent speakers who will follow me in this talk. Andy Knoll will, I'm sure, focus on paleontological themes. Pam Soltis will probably deal with phylogenetic themes as well as the digital topics she talked about. And Melanie Stiastny, I have no idea what she's going to talk about, but it'll be great, I assure you. Then I'll also, though, want to mention that there are other, especially more and more these days, natural history collections are finding additional use, not just in the traditional areas of systematics and taxonomy, but they're being applied to address contemporary problems in biodiversity and beyond, societal level problems. At the same time, there are worrisome and ongoing threats to the survival of natural history collections and museums, and I'll give you some illustrations of these. It brings us to a very ironic situation because this is happening at the same time natural history collections are becoming increasingly valuable, and there is so much promise being shown in their future directions, particularly the digital applications which are providing universal access to, and the use of collections and their metadata. So that's how we're going to organize this presentation. All right, let's start just a brief overview of collections. No discussion of the origin of natural history collections would be complete without a mention of the Wunderkammern, the cabinets of curiosities. This was a tradition begun usually by individuals, wealthy individuals, exclusively in Europe, who would amass their own personal collections through their independent wealth of particular, it was very idiosyncratic. It reflected their personal tastes or the access that they had to particular kinds of organisms. This is an illustration published in 1599. It's the interior of Ferrante Imperato's Museum and Library in Naples, Italy. Imperato was an apothecary who published a natural history book, and he included this illustration in it. Now, subsequent to the 16th century, moving on into the 17th, 18th, and especially the 19th, we saw a continued interest in global natural history and wonders of the world, if I can use that term, combined with greater professionalization of science and natural history as an actual discipline. And we see associated with this the founding of the great museums of Europe, natural history museums of Europe, which became centers of research and teaching, but also holders of vast collections for global biodiversity. This is one of them, the Muséum National d'Histoire Naturelle in Paris. This museum was formally founded in 1793 during the French Revolution, but it originated more than 100 years earlier when the collections were founded by King Louis XIII. Another example of this, of course, is the Natural History Museum in London. The building shown here, which is the one that you can still go to, still a public institution in London, was opened in 1883. The original collections date back to 1756 when they originally purchased for public display. Now, this was Europe so far. What about North America? Well, as you can imagine, as the colonies continued to develop, and especially after the American Revolution, there was greater interest in the discovery of the New World was expanding. There was great interest in establishing museums in their own right here in North America. There was also, to some extent, a matter of national pride or continental pride to show that we could have museums as great as those in Europe. I'll give you an example of that in a moment. Here's the first one. so-called Peale's American Museum. This was founded by Charles Wilson Peale in 1786, so shortly after the American Revolution, founded in Philadelphia. And here is an illustration or painting. This is actually a self-portrait by Peale, who was quite an accomplished portraitist, of showing Peale in his museum, which he painted in 1822. But perhaps the greatest example of a single individual amassing enormous collections to indulge his or her particular passion for natural history was the man I mentioned already, Louis Agassiz. Swiss-born, came to Massachusetts in the 1840s, was ultimately recruited to stay here at Harvard. He had grand ambitions. Here he wrote or testified before the Massachusetts legislature in 1857, "My great object is to have a museum founded here which will equal the great museums of the old world. We have a continent before us for exploration which has yet been only skimmed on the surface. My earnest desire is to put our universities on a footing with those of Europe or even ahead of them, so that there would be the same disposition among European students to come to America that there has always been among our students to avail themselves of the advantages of European universities." He was very clear in what his intent was, and he said this over and over again over many years, and he did his best to carry it out. And he did build a museum that had vast collections. Here's a picture in the lower left of the original building of the MCZ. This is actually still there. It's the end of the building over on that side. That's the view from Divinity Avenue. You don't see that view right now because the Harvard University herbarium obliterates this part of it here. I wonder if we can get the HUH taken out so we can restore the nice view of the MCZ. But here is what Agassiz's son, one of his sons, George, Alexander Agassiz's son, George, his grandson, wrote in 1936 in the history of the MCZ. By 1867, the first portion of the wing, begun in 1859, was inadequate for the accumulating mass of material that poured in from all sides. Lecture rooms were invaded until it was almost impossible to move about in them. There are accounts of Agassiz. He would routinely command or purchase specimens from all corners of the globe, and there was an almost ceaseless string of horse-drawn carriages that would come up on Oxford Street and unload their 55-gallon drums of museum specimens into the building, put wherever they could find space. It bordered on a mania, I guess we could say. Certainly, I guess a more polite term is the grand ambition. And this is also revealed in a reflection by Joseph Henry, who was the first secretary of the Smithsonian Institution. In 1873, January, Agassiz was to die less than a year later. The Smithsonian Natural History Museum was founded in 1846. MCZ was founded in 1859. So by 1873, they were still young institutions, so getting their bearings, and Agassiz was intent that they sort out their respective territories, and Agassiz dropped in on Joseph Henry in Washington, D.C., and this is what Henry wrote in his diary for that day. According to Agassiz, the MCZ is now the first zoological museum of the world. Its object is to form general collections of the whole world. He thinks that the Smithsonian and the Cambridge museums ought not to duplicate specimens, that the aim of the Smithsonian is the preservation of everything American. Basically, in short, what Agassiz was saying, you can take North America, MCZ will take the rest of the world. A modest request. And just finally, just as a little travel pitch for those of you who have never been there, probably the last grand museum that indulged the passion of a single individual is this one. This is the Rothschild Museum in Tring. That's Walter Rothschild riding quite an eccentric figure, riding a Galapagos tortoise that he brought over from the Galapagos to Tring outside of London in England. That's, by the way, he's riding it, and that's a stick in his hand with a piece of lettuce on the end of the stick. He's kind of dangling it to help have the tortoise walk in front of him. Rothschild his particular fondness was for birds, and he amassed enormous collections of birds. Ultimately, the Natural History Museum is still there. It ultimately was incorporated into the Natural History Museum of London. which maintains the Trink facility. But if any of you ever have a chance to go there, it's a marvelous facility. It's preserved to a great extent in its Victorian splendor. It really is a wonderful place. Okay, that's our quick tour through the history of natural history museums and collections. I should also just mention, I was showing you some pictures of the galleries and so forth, and you might be saying, well, why did I keep showing pictures of galleries and not the collections? For most of the history of natural history museums, actually until the Agassiz, Louis and son Alexander, collections were deployed in the main exhibit halls. It was an innovation of the Agassiz to actually have a separate synoptic collection on display for the public and relocate all of the research collections to other parts of the building. What I'd like to do now is switch to documenting the very important role of natural history museums in biodiversity inventory, which is one of the cornerstones for studies of evolution. And as a context for this, I show you this illustration, which was produced several years ago by Gary Rosenberg, who's a curator of malacology at the Academy of Natural Sciences in Drexel University, then of Philadelphia. And it shows for several particular groups of organisms, birds, orthopterans. Orthopterans are grasshoppers, locusts, cockroaches, and so forth, particular kinds of insects, as well as fishes and marine mollusks, the rate of accumulation of new species, or species that are officially named. Frequently, one hears the 18th century and the beginning of the 19th century as the age of discovery, something looking back. When in fact, for most groups of organisms, of these four, it's only birds that is the exception. The rate of discovery since the late 18th century, early 19th century, has, if anything, gotten faster. We today are in the age of natural history discovery and species inventory. Species have never been described at a faster rate than they are being done so today. And it also indicates how many species are left to discover and describe, because there's, with the exception of a few kinds of organisms, there's no sign of the curves tapering off. I think you'd be, well, on the one hand, you'd be surprised, maybe not so surprised, that it's not easy to get an indication of how many species there are named or how many there are being named in a given year. It's not an easy thing to do. All the digital tools that are being developed now to accomplish just that have not reached their fruition. This is the most recent account that I could find of the numbers of species described in a given year. It was published in 2011, actually for the year 2009. And here we have, for a series of different kinds of organisms, the number of new species of each of these groups described in the year 2009. We just need to come down to the bottom here. 19,232 new species of organisms were described in 2009, bringing to a total of not quite 2 million named valid species alive today. This was 2009, we're now in 2015. If we extrapolate this figure forward, there really is now more than 2 million species. It's quite a staggering number, happens quite commonly. So commonly, in fact, that new species are still being found in the places you'd least expect them. Here is a new paper describing a new species of frog from the remote mountain city of New York. This paper was published in October of 2014. Here's a quote from the article: that this discovery is unexpected in one of the largest and most densely populated urban parts of the world. It also demonstrates that new vertebrate species can still be found periodically, even in well-studied locales rarely associated with undocumented biodiversity. Here's a picture of the frog around a Kauffeldei. If you're curious where actually in New York, it's not found on 42nd Street. There are still breeding populations on Staten Island and elsewhere in New Jersey and parts of Connecticut and so forth. Interestingly, the biologists who discovered this were tipped off by the call. Males have a very distinctive call that is significantly different from other leopard frogs in the area. That he said, "Whoa, wait, what is that? That can't be a leopard frog." And sure enough, it was a different species of one. And its distinctiveness was subsequently confirmed by molecular techniques, DNA sequencing and so forth, which show that it really is quite distinct. The discovery of new species happens all the time, but what is even more surprising, perhaps, is that the location where these new species are found is, more times than not, natural history collections. Here is an article from the Washington Post. The press loves this stuff. It's somewhat surprising for reasons I'll mention in a moment. From just a month ago, January 21st, from a new bush cricket that was discovered, and it's actually several species of them discovered in museums in Europe, and recently described they had been collected many, many years earlier. Here's an article that appeared just a few days ago on the BBC website. This is a fossil, new fossil species of dinosaur that also, it says down here, fossils stored in Doncaster Museum for 30 years. It's quite embarrassing for the curators. They're thought to be a plaster copy. Has turned out to be a new species of ancient reptile. Another species that was sitting in a museum collection. And in fact, this happens so frequently that I think the press is really getting the story wrong. What they should be focusing on, what's news, is how common this happens. How many new species there are waiting to be described in the world's natural history collections. So the scientists have been documenting this phenomenon in the last few years. Here's a paper that was published 2010 with respect to herbarium collections, flowering plant collections. And they found a tremendous number of new species are being described from existing collections, and they're pretty old specimens. Only 16% of new plant species were described within 5 years of being collected for the first time. Nearly 1/4 of new species description involves specimens more than 50 years old. Of the estimated 70,000 species of flowering plants still to be described, and ferns and so forth still to be described, more than half already have been collected and are stored in herbaria. Now, I guess this is bad news if you're a new graduate student who's just come here and you see yourself as the next Indiana Jones. You want to go out to some really exotic, wild place to find new species, when the reality is, just take off your coat and just go over to the herbarium and you'll be able to find more that way. Now, zoologists have also tried to document this phenomenon, and it is true for zoological specimens as well. Here's an article that was published more recently, 2012, where they posit the concept of shelf life. That's the time when a specimen has to sit on a museum shelf or in an herbarium cabinet, how many years before someone recognizes it as a new species. Based on a random set of species described in 2007, across all kingdoms of life, we determined that the average shelf life between discovery and description is 21 years. The length of the shelf life is impacted by biological, social, and geopolitical biases. Okay. Now, I can actually give you an example of this from my own history. Now, which of these two people do you think is a fellow of the Royal Society of London? It's not that person on the left, and that's me as a graduate student in 1975 in Mexico doing my dissertation research collecting salamanders. And I didn't realize it at the time. Initially, I subsequently came to realize it, that I was inextricably linked with this man shown on the right. He's Hans Gade, a famous European explorer, naturalist, Cambridge Don, ultimately inducted a fellow of the Royal Society of London, who made a very long and famous expedition through southern Mexico, and he wrote a wonderful, just marvelous book, a travelogue chronicling his expedition through Mexico. It's a very thick book, and the reason I became inextricably linked with Gade is that here is a map of southern Mexico. This is where Gade went, and his book shows maps. He tells you basically every day where he was, all the villages he went to, every road he took, every burrow that he took, and so forth. And this shaded region here, these two areas, that's the range of a particular genus of salamander that I was studying for my dissertation, PhD dissertation. And my assignment, when I was accepted my dissertation topic, was to basically go everywhere in this area that I could find or get into, and if not collect every salamander that I found, at least find as many localities that had salamanders that I could find, and take samples to try to sort out what the species are. And it turns out I went to a lot of the places that Gade had gone. And one place in particular where Gade had gone, which was extremely important, was up here. It's Volcan Orizaba. Hard to read the writing on this. It's a large volcano, extinct volcano. Here it is, Volcan Orizaba. It's a very beautiful spot. This is the highest point in Central America. It reaches about 17,000 feet as its peak. And Gade went pretty far up onto the volcano, and there's nice forest up here, and he found the salamanders that I wound up doing my dissertation on. And I went up in the same places, and in fact went to the same villages that he had been. Now, when Gade was there, he collected some specimens from those forests and brought them back and deposited them in the Natural History Museum in London in about 1902, 1903. And there they sat for decade after decade. At the time, this genus, it's called Thoreus, there was only one species known, and that's the name that was put on it. Nobody bothered to look at those specimens again for a long time. I did my work in the 1970s. By the 1970s, here's a Thoreus. This gives you an idea of how small they are. They're very tiny animals. That's a blade of grass. Another 8 species had been described by 1970. So there were now 9 species known in the genus. And I did my work. I had the benefit of molecular techniques when you can identify species identities and relationships using molecular means. And I found there wasn't one species, there weren't 9 species, there were 25 species, and now we believe there are more than 30 species. And what I wound up doing for my work was describing several new species, and all of these 3 here are ones that I described, ones of several others. And in fact, this is one of the species that Gade had collected in 1903, and in fact, in our species description, we included Gade's specimens. In fact, it even got more interesting than that. It turns out there are 2 species found here, and he collected both of them. And had he looked carefully at them, he could have told them apart, I think, even without molecular techniques. So here's the paper that we published. This is way back in 1998, which is 95 years after the specimens were deposited in the Natural History Museum, London. They were finally given their correct name and described as new species. All right, species inventory is one thing, but there's a lot of bad things happening in the world these days having to do with biodiversity. Obviously, everybody here knows that. There's a biodiversity crisis underway. Many species are being lost. Many species are approaching extinction. Some of them are quite exemplars of the phenomenon. Here's one, the polar bear. And here, too, natural history collections can be of great use in, if not absolutely saving the species, contributing information that can be used, developing conservation strategies to help save biodiversity. Here's another iconic group of organisms with respect to endangerment and need of conservation. Those are the Galapagos tortoises. Galapagos tortoises, of course, were decimated by whalers who visited the Galapagos Islands, especially in the 19th century, early 20th century. They are now protected, but many of the populations were wiped out on different islands. A lot of them were jumbled and mixed up and so forth. And there's a tremendous mission now, an initiative to try to bring back the Galapagos tortoises and restore their native populations to their original sizes and help propagate them and so forth. And this is a study that was published a few years ago. I'm showing because it provides a nice opportunity to show another important aspect of natural history collections, which never was imagined when they were originally founded, which is their utility for studies of molecular biology. When DNA was initially discovered in the 1950s, I don't think anyone imagined that within 50 years, one would be able to go to a natural history collection and take an animal preserved in ethanol or formalin or a dried bone and pull out DNA from it and sequence it and find out about the genome of that organism. But that is what can be done routinely these days with great success. And it's been a real boon both to natural history collections and to those who wish to use them. We have here in the MCZ one of the few collections of Galapagos tortoises from really early days. And this is one of our specimens. This is one that was collected in 1835. This is a close-up of the shell. It was from the HMS Abigail, which sailed out of New Bedford, Massachusetts. It came back, this ship came back, brought this specimen with it. It was given to the Boston Society of Natural History. Eventually, the Boston Society of Natural History closed. It and several other shells made it into our collections downstairs in the building right here. We had a visit from Gisela Chacon from Yale University several years ago, who was doing a project on Galapagos tortoise conservation, and she was interested in resolving the evolutionary or phylogenetic relationships among the Galapagos tortoises to try to help plan their conservation strategies. And she asked if she could take some bone samples and pull out DNA from it. And so we said yes. She came up, kind of an unusual tool for a molecular biologist. She brought a drill with her with drill bits. And she just took small samples from many of our tortoises, and they successfully pulled out DNA from our tortoises, which turns out were indispensable for completing the analysis. Here's an illustration of some of their results. First on the left, this is a map of the Galapagos Islands. The islands shaded in gray still have viable turtle populations on them. The islands that are open, unshaded, no longer have any turtles on them. The capital letters are the island names. The italicized names below are the names of the different tortoise populations. Originally, the Galapagos tortoise was believed to be a single species, which had a series of subspecies. More recently, biologists interpret them as being viable species. Each island basically has its own species distinct from others, although in some cases, for instance, in Isabella, a single island may actually have several different species of tortoise, sympatric, on the same island. And one of the results from Chacon's work was that on the island of Santa Cruz, which is here, which is shown in an exploded view here, there are several populations of turtles, 3 in fact shown here. And if you look here, it's hard to see, I appreciate, these are the shells. The turtles differ in their shell shape characteristically. At one time, they were considered all the same subspecies or species. But in fact, using the DNA samples from blood samples from living turtles plus ours, she found that, in fact, that population there is another species, undescribed species of the Galapagos tortoise. But more interestingly, this population, these are our turtles. The Floriana Island samples, of which there are no more turtles on the island, is an undescribed new species. We have samples at the MCZ. The bad news is that apparently it's extinct before it was described. Now, I say apparently because an update, and this is more recent, having done all this genetic analyses, they've been surveying turtles on other islands. And recently it was discovered that a population of tortoises up here on northern Isabella Island has genetic signatures of Floriana tortoises. And they've gone back and they've looked at the ship logs to see which way the ships were moving. It turns out that it was very common for whaling ships, to take on live tortoises here at Floriana and continue on to Isabella. And what seems to have happened is that the ship came here and they were taking on more supplies, and the sailors realized, oops, we don't have enough room on the boat for these other supplies, and they unloaded turtles from Floriana onto Isabella. And they have found some hybrid animals. Apparently these animals then bred with ones on this island. And they're now going around trying to census every turtle on that island and doing genetic identities, because there is a chance, these are animals that live a long time. There may still be some purebred Floriana animals wandering around on this island. But none of this could have been revealed if they hadn't been able to get DNA out of our museum samples. Quite an unlikely story, but a really marvelous one. Okay, other areas in which natural history collections are being used to address conservation issues or biodiversity crisis. This is one that's particularly important to me. I'm an amphibian biologist. I suppose many of you, if not all of you, have heard about the global population decline of amphibians. It really is quite serious, and it's reached great epic proportions and has achieved great notoriety, as seen in this cover of the New York Times Magazine. It's really quite horrifying to see what this is about. Here are some frogs photographed in the Pyrenees Mountains in 2010. They are dead. They were just found like that on rocks. And it turns out, once they were analyzed, they have a fungus. They succumbed apparently to an infection by the so-called Kitrid fungus. Now, Kitrid die-offs like this were discovered beginning in the early 1980s. The fungus, the species of fungus that actually is associated with it, was not discovered or named until the late 1990s. when people finally had some potential cause to implicate as the reason why this was happening. These die-offs caused by or associated with Kitrid fungus have now happened all over the world. There are a few places in the world where it hasn't, but generally speaking, it's quite widespread. And thanks to museum collections, people have been able to identify the chronology of these infections and identify where they happened and when. Here's a pretty terrifying illustration. This is now, we've moved from the Pyrenees to Central America. Here's Costa Rica, here's Panama, Colombia is over here. This shows the so-called wave of death of the Kitrid fungus, which apparently was first identified or first invaded up here in Costa Rica and has gradually spread over these intervals in further south and ultimately into South America. Each of these circles is a known die-off. And in many of these instances, people have gone back to museum collections, and you can find the Kitrid fungus in specimens collected at that time, ones that didn't succumb to it, collected at that time, either because their spores can be found in the skin just by simple histology, or by using now molecular techniques where you can simply swab the skin of either a live amphibian or a dead one and find the DNA associated with the Kitrid fungus. So similar phenomenon is shown here. This is a more recent one. Beginning in June 2013, divers in the Pacific Northwest were finding die-offs of millions of sea stars, of more than 20 species. And it's caused by a disease called the sea star wasting disease. Here's an illustration of a healthy sea star. And here's a sea star that has just begun to develop the wasting disease. It's called wasting disease because it infects them. The sea stars become listless. The disease spreads out over them. They die, and they just kind of develop into ooze, waste away. Smaller die-offs, though, were known since at least 1979, but otherwise it came out of nowhere. Just in the last couple of months, a paper was published that identifies the cause or implicates the cause of these die-offs. Not a fungus, but a virus, the so-called Denso virus, which is associated with sea star wasting disease. There's all kinds of aspects associated with this, but the authors were particularly interested in going back to see, once they were able to identify a genetic signature for the virus, going back into museum collections to see if they could, how far back in museum collections they could find it. And they went to the Los Angeles County Museum in Los Angeles, which had sea star collections going way back, and they found that they could detect DNA from the Denso virus as early as 1942, suggesting, in those same localities where the die-offs occurred more recently, suggesting that the virus has been present in the North American Pacific coasts for at least, well, more than 70 years. Now this, of course, raises the question, why now? If this potential disease was harbored for decades, what has happened? Is it pollution? Is it climate change? Who knows? But this at least, again, it's be overstating it to say this in any way offers a solution, but at least helps us to diagnose the problem and ultimately may contribute to some kind of a solution to treat this horrible disease. Okay, so those are several examples of how natural history collections can be used of their importance. You'd think with all of this great stuff that natural history collections can be used for, natural history would just be constant joy and mirth in natural history museums around the world. Not so much. In fact, over the last 10 or 15 years, there's been periodic stories about various plights faced by natural history museums. Typically involves finances, but not always. Here's one from the Great Field Museum in Chicago. Has had some very hard times in the last few years. They've had to lay off much of their staff, the scientific staff. That is from January of 2013, a couple of years ago. Here's a more recent one, the Carnegie Museum of Natural History, another great museum in the United States, in Pittsburgh. This is a press release or an article from November of 2013, a little more than a year ago, offers buyouts to their curators. More recently, just at the end of 2014, Italian Natural History Museum is on the verge of collapse. This is a European phenomenon as well, and in fact, even as recently as last week, here's an article in Nature magazine not just about endangered organisms, but about endangered collections and endangered museums. So it is a difficult time, and it's a great irony and in some respects a tragedy that many museums and collections are having so much trouble justifying their existence, or at least keeping afloat, despite how valuable collections are. I don't have a silver bullet to offer you or surprise you with, but I will say it's not entirely bad. There actually are some good news on the horizon, because the value of natural history collections is, I think, increasingly being recognized, and being recognized by people who count. Here's a somewhat of a remarkable letter that I was sent, not just me personally, a memo from last March, published from the office of the president. Now, this is not the president of Harvard, of course, but it's only the president of the United States, but still, okay. This is authored by John Holdren, President's Science Advisor, championing the value of scientific collections and giving, to some extent, marching orders to the federal science collections. And in particular, it mandates them, or the subject is improving the management of, but particularly access to, scientific collections. Because one of the things that's holding back the utility of natural history collections for researchers, for teachers, for conservationists, is the material, the information in them is often difficult to access and to use. And it's a very proud statement that this memo starts with. Scientific collections provide an essential base for developing scientific evidence and are an important resource for scientific research, education, and resource management. Scientific collections represent records of our past and investments in our future. They are also tools that can be harnessed to address challenges facing humankind. Pretty inspirational. Now, this didn't come out of nowhere. In 2009, this document was published by a group of an interagency working group. There has been a lot of people within the US federal government championing scientific collections, trying to get them greater attention and greater support. One of the manifestations of this is a great resource, which I suspect, because it's so new, most of you may not have heard of. It's called Bison. Not bison as in buffalo, but Bison as in biodiversity information serving our nation. It is a website maintained by the US Geological Service. It was just released a few months ago. And it basically provides access, or will ultimately, to all natural history collections and observation occurrence data held by federal agencies in the United States. It's user-friendly. It's designed to be used by the public. You can go to the site. You can download information on the US overall. You can download it for individual states, for individual locations. You put in particular species that you're interested in, and it generates maps. Here's, as of I just checked this today, as of February, earlier this month, more than 200 million observation records can be accessed. Of those, almost all of them are georeferenced, meaning you can plot them on a map. And there are more than 300,000 species represented of the 1.9 million species on Earth. It's really quite an impressive resource. It generates maps such as this. Distribution map for the mountain maple, a species found in the northeastern US. It's a cool adapted species. These green dots are museum records or herbarium records. These blue dots are observational records. And you can see where the species is found. You can overlay it, as is done here, on different climate variables. In this case, this is the July annual mean daily average temperature, which is, of course, cool in the north, warmer as you go south, hot as you go further south. You can see that this is a species that likes cool forests. And in fact, even when it gets into the southern regions, it does so by going down through the Appalachians, where it stays in the cool forests. You can choose particular dates of locality records. You can compare species distribution in the early part of the 20th century with the later part. It's tremendously versatile for all kinds of uses. That's federal collections. What about non-federal, such as the MCZ and other university and freestanding museums? Well, we've been organizing as well. You have to publish your own government booklet. So here was ours that we published a couple of years ago, championing this, the Network Integrated Biocollections Alliance. This is an aggregation of natural history museums, and it too has generated tremendous amount of support. It led to an initiative sponsored by the National Science Foundation to the tune of about $100 million over 10 years to found a linked network of natural history occurrence data from biological collections in the United States. It's maintained at the University of Florida. It's called iDigBio. It has 26 million records, including those from the MCZ and a lot of other institutions. In fact, 373 different institutions. What's more exciting, it's more versatile than Bison. It links the collections data to information about evolutionary relationships, ecology, paleontology, living collections, links to genomic data, to living frozen tissue collections, and other repositories. An extremely exciting initiative because all of these disparate data sets are being linked together, and you can combine them in very novel and important ways. Let me just give you one example. You can use these data to look at predict effects of climate change. Let's just look at this lower sample. These are illustrations for this species. This is Prunus geniculata. The scrub plum is an endangered species of plum, native to Florida and endemic to Florida. It's a federally listed endangered species. This is its distribution now. There's a series of white squares here, and it's showing most of the distribution is within this green zone. What you have here, these colors represent a series of environmental variables that represent features in the case of the green that the species likes, and it's found there, and other combinations of environmental variables, temperature, humidity, and so forth, that are not as favorable to this particular species. So now the species is found within that green envelope, as well as where other places that it exists. Well, climate change is upon us, and people now are able to make pretty reliable predictions of how climate for particular parts of the Earth are going to change from 2015 to 2050. And here's how the distribution of these environmental variables will change in Florida from now to 2050. Where is the green? Green has disappeared. Instead, it's all red. The climate has dramatically changed. So what is a very reliable prediction is this endangered species will completely disappear from its current range, or its current main range. There still are other green areas here and there where the species presumably can survive, but it either has to get there on its own, or we humans have to establish it there with some assistance. These kinds of analyses are made possible by taking natural history data, putting it online, and plugging it into these models. Other uses have to do with natural history data from collections in studying phylogeny. There are now all kinds of cool digital tools online to incorporate natural history collections data with phylogeny. Here is one called OneZoom. Here's one I like even better called PhyloGive. This is created by the Atlas of Living Australia. Here's a so-called phylogenetic tree or a cladogram. This is the bread and butter of people who study evolutionary relationships. This branching diagram shows the relationships of different species. Each of these names here is a species of acacia, kind of bush or tree, found in Australia. And when species are closely allied, that means they're each other's closest relatives, and then they hook up to other more distantly related species. These have been around for many years. What is new is that you can now take your results of a phylogeny, press a button, and it will generate for you a range map color-coded by the particular colors on your phylogeny, and you can see the geographic distribution of species on your phylogeny in real time. So for example, for this species, Acacia cressocarpa, here's the color code. There it is up here. You can compare it with its sister species down here, which actually is distributed over here. Some theories would say a sister species, a closely related species, should be found in the same geographic area. Other theories predict that because two species are so similar, they would not be found in the same area, and that actually seems to be the case. But you can do these things. Before, it would take maybe hundreds of hours as an exaggeration, but many hours to do this kind of thing. Now you can literally do it in seconds with the information that's available online to study evolutionary relationships. And then ultimately, you'd like to have all of this natural history information generated for individual countries linked together. And in fact, it is. an entity called the Global Biodiversity Information Facility, takes the data from all these disparate sources, Atlas of Living Australia, Bison, iDigBio, combines them. As of this morning, there were nearly 527 million records available. Here they all are. The red is a record, and it lets you see immediately some parts of the world, either where they've never been collected very densely, or for whatever reason, the countries have not been able to, the institutions have not been able to put their records online. But again, it's a tremendous resource. Now, I'm going to end now and come back to this man, Tom Barber, who I was kind of disparaging of. I told you some embarrassing stories about Barber and collections, but I want to show you a more positive side of Tom Barber and show you another quote of Barber's that he put in the MCZ's annual report for 1930-31. In no way can so wide a grasp be gained of many aspects of animal life, and above all in the appreciation of the processes of evolution, as by working in a museum in daily intimacy with its great collections. The museum is no mere storehouse of dead and dusty trash, but is a living and vital center. The museum's place in present-day biology grows more, not less, important as its collections grow. And I would like to come back also to Henry Rozowski. I have great respect for the man, but I guess we can say, no, maybe we can forgive him for not envisioning the day when molecular biologists, if not clamoring to use natural history collections, would be aggressively approaching curators in natural history collections all over the place for access to their specimens, particularly access to their tissues for their molecular studies. When molecular biologists and biologists of many different kinds would be seeking collaborations and means of using natural history collections data. It really is a new world that one couldn't have expected. And so, with all due respect to Mr. Rozowski, not true. Natural history collections have a very promising future, and they have a very vital role to play, and I look forward to seeing it with you. Thank you. So we have time for, where's Jane? We have time for questions? Yes, I'm looking at Jane. So please, if anyone has a question. Yes, please.
Well, first of all, I'm really happy that you had some good words for Louis Agassiz, because I think he just showed that even though his ideas were wrong, the things that he collected really, really have spoken for themselves over the years. And that's such a wonderful thing.
Yeah, I don't know if you heard the question. Louis Agassiz, you said Louis Agassiz was wrong, but his collections have really proven the test of time. Actually, I'll be even more favorable to Louis Agassiz. It's easy for us in the modern day to look back in the 19th century and say who was right and who was wrong. Louis Agassiz was a tremendous observer of nature. He did marvelous work. Yes, his theory ultimately was proven wrong, but as a scientist, he was tremendous. And regardless of that, his collections, he really had the right idea to amass collections, which have proven to be tremendously important. Yes?
I have a question about history museums, the historical part of your talk. Why weren't Agassi and Barber, they were both involved with the Boston Society of Natural History. Were they kind of neglectful of the BSNH, or were they kind of, it seems like they had their own agendas with, Agassiz, his own museum.
The question is, why weren't, paraphrase, Barber at the time, and others, contemporaries, more supportive of the Boston Society of Natural History and let it go under? Actually, truth be told, I don't know that much about the Boston Society. David, do you have anything to offer? We have a local museum expert here in our midst. I'm going to put him on the spot.
It's a long story.
I think also, though, as being a museum director, I'm sure Barber felt to some extent the same way. I've got my own set of headaches to deal with. And so maybe he felt that if he could do his best to keep the MCZ going, that was enough on his plate at the time. Maybe that's not doing proper service to what happened, but I don't really know. Yes?
What kind of rules have you had to establish with all these
That's a great question. It's a tough, I have to be careful how I answer this. Because on the one hand, the basic priority or imperative of a museum is conservation of the specimens. At the same time, we are a research museum. And it is also an imperative that we try to make our material available to be used. So not surprisingly, we love what's so-called non-invasive sampling. And that's why we love X-ray machines and CT scanners and things like that. Now, for some, but yes, there is some destructive sampling which has to be done. Fortunately, for a lot of DNA work, the amount of DNA that you can get away with and get your sequences is ridiculously small. So if you have a reasonably sized specimen, nobody would even notice if you, and sometimes, if it's a frog, they can take a piece of the liver or something. Nobody's going to ever notice that it's gone and this kind of thing. There are problems when the specimen is much smaller. And also if it's a so-called type specimen. There are some specimens which are the exemplars, the name-bearing specimen. And holotypes, for instance, each species of animal has a holotype, a specimen that is associated with the name. Holotypes are supposed to be preserved totally untouched. Occasionally, for very extremely rare species, all we have is the holotype, and we get requests. We deal with those on a case-by-case basis. We just got a request today for an instance like that, and we didn't say no, but we said, you need to show us that you can do your processing properly. In other words, if we gave you the sample, it wouldn't be squandered. So it has to be done on a case-by-case basis. Some museums are more stringent than that, and they just say, sorry, no. But my personal view is, again, we have to balance the preservation and conservation imperative with the need to make the stuff useful and usable by people. And it's not a big problem in terms of, say, most times you can deal with it, and it's really not an issue. Yes, sir, Mr. Suit.
I would like to ask if you could comment on Agassi and his anti-evolution point of view, while at the same time there was so much evidence coming out of geology, about the strata, older species, the type of species at different age.
Yes.
Geological strata. And was he unaware of what was going on in geology? Because it seemed like such a smart chap.
That's a great, Dr. Suit's question is, can I try to reconcile Agassiz being an anti-evolutionist with the seemingly obvious signs in the fossil record and in geology? And was Agassiz ignorant of all of that? Well, the answer to the last part of your question is definitely no. Agassiz was a professor of geology. And in fact, Agassiz is to some credited by many with having discovered, among other things, the Ice Age. And he was an extraordinarily accomplished geologist, in Europe especially. He was very aware of the fossil record. He documented what we would call the fossil record today. Where he differed from Darwin is not on the observations, but the interpretation. He believed in a series of floods. I mean, he believed in a more biblical, literal interpretation of the Bible. And that explained, he just simply believed that he didn't deny you had all of these things, these specimens in sequential time periods, but he just simply refused to believe in transmutation, that one evolved from another. I can't explain it totally logically, because I don't think it's totally logical, but that's what he believed. He was by no means ignorant, though, of the fossils and geology. Yes, sir.
Is there any collection in the world of the microbes, bacteria, viruses?
Is there any collection of microbes, bacteria? The answer is yes. I don't know that much about them. It's mostly, as you might expect, in a medical context. Where is Nomi? Nomi, do you know the answer to this one? Can you help me out here? Okay. I'm punting here. Yeah, so it's mostly in medical-related institutions. It's not a very sexy kind of collection. You know, you can't have museums that wander under the skeletons of the giant bacteria the way we have here and so forth. That is, yes, that's where the action is in terms of new species and so forth. You're absolutely right. Yes, ma'am.
You mentioned a little bit about the virtual sharing of the collection. Many collections, not only just scientific history collections, are doing three-dimensional scanning of their collections because some of this natural history material doesn't last forever. Are you beginning processes of that within this collection?
Yes, question is, are we doing, or institutions like ours, doing 3D scanning? And the short answer is yes, a tremendous amount. One of my regrets when I put this talk together, I realized I was already full and I couldn't talk about imaging in any great extent. We are doing it particularly in a lot of other places in terms of 3D CT scanning in particular, but also surface scanning. And this is going to explode in a short amount of time because obviously with 3D printers, one can soon, in fact, I was contacted last week about MCZ developing our copyright policy for this.
That was the other question. What, if any, do you have on the kind of the three-dimensional virtual object sharing, lending, what are the rights to those things? And it's a question in a lot of fields.
Absolutely. I can only get, it's evolving. In general, we, and this is, I guess, Harvard, I've been encouraged also by the administration, and I personally believe this as well, we believe that we should share our information as much as possible. So when we get copyright requests, just even for standard publication kinds of images, if it's a non-commercial publisher, we give it absolutely for free. For scientific use, we just routinely do this kind of thing. For commercial publishers, we'll charge a small amount and so forth, but we feel we should be sharing this information. How we're going to deal with 3D images. Actually, there's a branch of copyright lawyers that believe once you take a 3D surface from our scans, we no longer own the copyright. In other words, that's a kind of a secondary derivative of it, and we can't control it in any way. So it's being worked out. And my first question when I was asked last week, said, we need to come up with this policy. He said, oh my God. And I said, what are other museums doing? That's my, because I figure, has anybody figured out to solve this one yet? And the answer is not really. Okay, maybe one more and then.
We have time for one more.
Okay, yes, sir.
It seems to me that your talk is a plea for the material specimen, material reality that comes into conflict with the digital reality. Now, you get out of it one way, say the molecular students have to go back to the specimens. The scanning and the imaging is all going to improve and improve and improve. And it will possibly get to the point where, like in the humanities, there was a really aggressive article a couple of days ago in the Times about how the humanities are going to disappear. Nobody's going to read books because we have a statistical study of books that are going to be so far superior.
Kind of like the Cliff Notes, for digital Cliff Notes.
But at what point does the material, is it dried out? The material is dried out and there's nothing more in there. In science, maybe that's never. What else?
That's a great question. I'll try to give a brief answer because I know Jane wants to wrap this up. We believe in the real deal, the real item. That's what we're charged with. However, in order to make as a means of making the real deal as accessible and available as we possibly can, we also champion virtual equivalents and so forth. Now, nobody knew, as I said, when 100 years ago when they put specimens in ethanol in the MCZ, that 100 years later or 75 years later, people would be able to get DNA out of them. Now, maybe this is kind of a cheap way of getting out of your question. There may be future uses for the real specimens or information that we don't even think of now, that somebody will come up with a means of extracting. So I think it's important to keep it. We are committed to the material item, but we try to, again, maximize the access to it, the utility of it by virtual means. They're not incompatible, but it does, to be sure, making a lot of the questions that researchers want to answer from our specimens, they now can get from a high-quality X-ray or remote information, we don't need to send them the specimens anymore. That is true. It's having that impact. Okay.
Thank you very much.
Okay, thank you.